are still dealing with genetics, but we are shifting to beyond Mendel, right? We did Mendel, and then we got into a little bit of the genetics behind it with meiosis, and you took an actual very simplified being through meiosis and created gametes to show how that process actually works. Now we break into, first of all, kind of growing beyond Punnett squares, to where you're not going to need Punnett squares anymore to do genetics problems, because we're about to get into some genetics problems that don't follow the rules that Mendel laid out. We're about to break some of those rules because some genes don't work exactly like simple dominant recessive, two alleles to choose from. We're about to sort of systematically break those rules to show you some of the exceptions to what Mendel said. Before we do that, we got to talk a little bit about, well, first of all, a term you already have heard, I'm not sure you've seen, karyotype. That's the one where, you remember Morgan, the fruit fly guy, where his grad students were like, hey, you don't have to sort the chromosomes physically with a stupid little needle. Just take a picture of the cell, cut the pictures of the chromosomes out, right? And then sort them by size, and you saw that. And at this point, we know quite a bit about which genes are on which human chromosomes, to where you can basically look up each of those 23 chromosomes, because remember, there's really only 23, right? You have 46, but it's really 23 pairs. Each one of the 23 has its own genes, but the two versions of that chromosome you have might have different versions of those genes, called alleles. Matter of fact, if you ever have to be tested for genetic disorders that might be suspected, they think you might have, a lot of times what they will do is a karyotype. And that's why I happen to have, you've seen the photographed karyotype I have of a former student. Because the banding patterns on your chromosomes when they do a karyotype, there are actually things they can look for that a trained genetic counselor can actually find and say, hey, it looks like maybe there's this allele, this disorder, this whatever. So here's a real limited list of some of the things that have been mapped to human chromosomes. Want you to, when you get it, when you're done writing, just look it over. Just kind of look through some of them, see what jumps out at you. Students always have questions. So you can see 22 chromosomes numbered. And then, of course, that last pair is weird. That last pair, there actually are different genes on the two members of the pair if you are male. So they're showing the X and they're showing the Y separately. What do you see that you want to know about? Yeah.
Taste receptor.
The taste receptor, or a taste receptor. Which chromosome?
1.
Yeah, so that's like one of those things on your tongue that allows you to taste things. Now, I'm not sure if it's one particular kind, whether it's the sweet or the sour or the bitter or whatever, or if it's just a generalized, that's what produces taste receptors and then they get changed from there. But yeah, there's a gene that codes for taste receptors. Yeah.
See, there's like alcohol, dehydrogenase, and marijuana receptor.
Interesting. Yeah, both of those, well, they're different. Alcohol dehydrogenase is the enzyme you make that breaks down alcohol. So variations in your genetic code for that can make someone more or less susceptible to alcohol, right? It's been tied to alcoholism, because if you happen to be very, very, very good at breaking down alcohol, those are people who might drink more because it doesn't have as much of an effect on them. So that's that one. The marijuana receptor is weird though, right? So you know about receptor proteins, right? This is one of those. Receptor proteins are like the ones you smell with, where there's some specific thing plugs into the receptor, fires some change off. In this case, that receptor happens to fit perfectly with tetrahydrocannabinol, THC, right? The psychoactive stuff in marijuana fits in there and fires off a bunch of changes, right? Which are poorly understood, but definitely make people feel different, right? And act different. Question is, why would there be a receptor for that naturally in your cells? For something that's an external substance that we, that grows or that we produce. It turns out for pretty much any drug you can think of, that is the case. What's going on there? Anybody want to know? Anybody think they know? So basically, it's a mimic, right? Basically, what that means is you make something naturally that fits into that receptor. You make something on your own that fits into the marijuana receptor, that fits into the various things that other drugs. Drugs are just chemicals we make that mimic body chemicals. What's really interesting, the one thing I know about the marijuana receptor that's super interesting, and I don't know how much you know about the effects of marijuana, hopefully not very much at all, but one of the things that it tends to do is screw with your sense of time. People report that time sort of slows down, that they don't remember what happened before, that they don't care what's coming, right? That it's just kind of, oh, okay, I'm here, right? So one of the places they found the highest density of marijuana receptors is the human uterus.
How does time and that, how, can you elaborate?
Can you think of a reason why someone going through childbirth might benefit from only living in the moment, not really remembering what just happened, not worrying too much about what's coming?
And also forgetting. There's a certain chemical that gets released when a woman gives birth that makes her completely forget about the pain. That's why when people say, well, if it's
I am here to,
why do you want to
Riley, oh my God, like you're preaching to the choir here. Like my wife is like, oh no, I love being pregnant. I'm like, you did not. You complained all the time. It was terrible, right? No, I remember it being really nice. That's because you're, right? The hormones make you forget. All kinds of, there's a whole system like that makes you, evolution has conditioned us to want to keep having babies, right? That receptor is part of it because it gets you through labor, because it helps you live sort of in the moment. At least that is the kind of current hypothesis about it. Marijuana is poorly studied and whatever. But yeah, interesting stuff. Riley.
Got to do with that.
Okay.
There's I see taste receptors, blood types, eye color.
Yeah.
But there isn't vision.
So
different with,
you're going to be disappointed in the answer. The answer, first of all, is that this is a really incomplete list.
Okay.
This is just a few genes that we know of, so that you're not overwhelmed. But the other answer is vision is there. Let's see. You're looking for rods and cones here. Where's it at? There's a blue cone somewhere. Seven. Yeah, blue cone pigment is that's one of the things in your eye that helps you see blue, right? Rods and cones are the receptors in the light receptors in your eye. So yeah, there's a couple there, not all of them, but a couple are listed.
My question was that would it be different for someone who's vision impaired,
Absolutely.
or someone who's blind or something?
Depends on the type of blindness, the type of vision impairment. Color blindness for sure. There's a very simple genetic mechanism. Sure.
How you see color, right?
No, the UV one might be a skin thing, but I'm not sure.
Oh yeah.
It might be. Yeah. Armani. Hexokinase is an enzyme that I believe that breaks down sugars. So it has to do with digestion. I think that's don't quote me. Yeah. Leptin is a hormone that has to do with your feeling of fullness after you eat, believe it or not. So the reason they've mapped that is, because people are different as far as overeating and stuff like that. So a lot of these, you're going to find on stuff that's been mapped, there's a lot of disorders. It doesn't mean that your genes are just out to kill you. It means that's the first thing we tend to study, right? Is if there's something wrong with somebody's genes, we're going to map that gene and see what it is. It's not that that's a gene for that disorder. It's just one where sometimes there's a mistake and it causes that disorder, which is probably why they've mapped the leptin gene, I guess, in probably studying like obesity and stuff like that.
In gene 4, it says red hair color. Is it?
Uh-huh. That's it, man. You found it.
What did my genes do wrong?
Or right. Come on.
Yeah.
Yeah. Now, it's so there's, I think if hair color is like eye color, I think it's like there's a brown and a not brown for one gene. But if you have two not browns, that's recessive. Then if you've got the red one, it turns your hair red, or if you've got the blonde one, it turns your hair blonde, I think. Yeah.
I see multiple different blood types. Like when how does like that work with if there's multiple different blood types? Does it like combine?
So there's one for the ABO, right?
Yeah.
And where's that? Number 9 has ABO blood group, but then is it RH factor somewhere? Number 1. Yeah, so those are actually two different genes that make it. So the RH part is the positive or negative. ABO is like your A, your AB, your O. And so those are the two probably most important medically. There's other blood stuff in here, but those are the two that go into what we call a blood type. And I promise you, by the end of this unit, you'll know how to do genetics problems with blood type.
What's Lorenzo's order?
Oh, you got me. I know it's a disease, and yeah, I don't know the story. We can look it up later. Yeah.
So like on some of the chromosomes, like 17 and 19, there's just a giant list of them.
Yeah.
But then on like 21 and 10, there's only one.
Again, it just has to do with what we've studied and what we haven't. And if you're really interested, that cabinet right there is the same thing you have in your hands, except with like, 1,000 instead of 100 different things that are in there. So take a look at it when you get a chance. So that's that. I just kind of wanted you to get a look at some of the things we have mapped and that we know about. All right, shifting gears again to how, and these are what, karyotypes look like. This is the one of my former student, which you can really only tell one thing, which is that it's a boy, because it's the X and Y at the end there. Pedigrees is where we're going now. Pedigrees are kind of the new Punnett square for us. Punnett square started so last week. Raise your hand if you remember pedigrees, if you've learned about pedigrees before. It's a really simple way to do a family tree, basically, with some very standardized, simple symbols. And those symbols are square is a male, circle is a female, a horizontal line connecting a square and a circle. What's that? Huh? Yeah. Yeah, right. We used to call it the marriage line, but of course that's not necessary to reproduce, right? So let's just be real PC and call it childbearing union. And if you drop a line from that horizontal line vertically down, and there's some more squares and circles coming out of it. Oh, I am, or the kids. You want to be careful, though. You want to be careful that you're drawing what you mean to draw. When you drop those lines, you want to make sure that angle is there, right? You don't want to like drop the line and then accidentally do this. Because that's not the story that you want to tell. That's a different story, right? That's illegal in every state. Yeah, good. We'll get there. There is a story about that we're coming to when we get into the royal families. That was kind of their thing. You get it? Pedigrees? That's okay. There's one more trick, and the only other trick is sometimes, well, most times when we do a pedigree, we're trying to track a particular genetic trait, and so we will shade in the squares and circles where the trait is, right? If we're tracking a disorder, we'll shade in the kids that actually show the disorder. Sometimes we'll give you a half shade. If it's a recessive trait, we'll give you a half shade to show you who the carriers or heterozygotes are. Most of the time not though. Most of the time. Only the shaded ones are there, and you got to figure the heterozygotes out. That'll be the case with the royal families when we get there. All right, let's practice. Flip your karyotype sheet over. And let's do numbers 1, 2, and 3. Which give you a pedigree. Like I said, we've got the shaded ones for, in this case, we are tracking dimples. And there's a little story about the pedigree, about Jane and Joe and Clarissa and Grace and Jorge. And so all you got to do here is read the story and write the name of each person below their symbol. So let's go ahead and do that right now. So I know you understand this, and then easy enough, right? You got it? So who's this? So that means these are. So if you were drawing this, reading the story, you would have started with Jane and Joe, right? So this is Jane. This is Joe. This is Larissa. Last year. Joe's dad has dimples. This would be Joe's dad. His mother. And Grace. And sister? Don't. James, Mr. Ronaldo. Okay, I don't know why we're naming him, but not them, but Mr. Ronaldo. And her brother, Jorge. Yep. And her sister, Emily. But her mother does. Will remain nameless. Got it? Okay, the rest of this is homework. Basically 2 more pedigrees to do. And on the last pedigree, which is my wife's family, you gotta do some eye color genetics to figure out what their genotypes are. Okay? Good. That is your homework due tomorrow, along with baby elves. Yep.